Could you tell us what sort of work you're doing to identify genes that promote myelin formation?
Sure, yeah, so that's the majority of the work we do in the lab actually, and it follows on from your previous question on fin kinase. So we really want to identify genes that only function in myelination. So the way we do that in the fish is we take advantage of the fact that you can interfere with multiple genes in fish. You can do so randomly, then you can look for effects on myelination, and thereby afterwards use genome sequencing to find out what genes specifically affect myelination. We can also take advantage of the imaging qualities of the zebrafish to look at precisely how different genes affect myelination and how those same genes might affect other cell types, so that we can really identify specific genes that regulate myelination. And then we'll take those on to studying their roles in remyelination, and with our colleagues looking at their roles in human stem cells and in mammalian models and so on.
That's very interesting, and I was wondering whether you could explain why using fish models, and in particular zebrafish, is advantageous as compared to other methods.
Right, yeah, no, that's a great question. One of the advantages of using zebrafish for doing these large-scale gene sort of identification screens is because you can collect a lot of zebrafish. You can, in our aquarium here in Edinburgh, we've got 4,000 tanks of zebrafish. It's very difficult to maintain other mammalian organisms at that level. We can generate lots of different fish that have mutations in lots of different genes and analyze their effects on myelination very rapidly. Myelination occurs sort of early in the development of a fish, so we can look within a matter of days how a specific gene affects myelination. To do so in other organisms would take much longer periods of time, and it's essentially impossible to keep that large number of animals and sort of identify genes in an unbiased way systematically. Of course, other organisms have advantages too, where you can interfere with individual genes, perhaps in a still yet more elegant way than is possible in zebrafish, although our technologies to interfere with specific genes is improving as well.